Hi, everyone. Welcome to this afternoon's session on our Neuro Lightning Talks. My name is Amanda Crocker, and I'll be your moderator for the session today. And each presenter will have 3 minutes to give a brief overview of their research. First up, we have Emily Riedick from the University of Rhode Island.
Hi, thank you. Cerebral palsy, CP, is the most common motor disability in children. CP typically involves muscle spasticity and is caused by perinatal injury to parts of the brain and spinal cord that are involved in motor control. Prenatal injuries capable of causing CP include decreased fetal oxygenation. Most individuals with CP experience chronic pain, which is largely considered a consequence of musculoskeletal abnormalities. Meaning that pain in this population is often dismissed as being caused by things like joint stiffness. However, we suspect that increased pain may be a byproduct of the neural injury itself. And we are investigating the link between prenatal hypoxic injury and pain sensation in a rabbit model of CP, which is generated by temporary occlusion of blood flow to fetal rabbits. Newborn rabbit kits that experience hypoxia in utero develop motor deficits that are characteristic of CP. Notice the wrist flexion in this picture. Next slide. We hypothesize that the prenatal injuries that cause CP, like intrauterine hypoxia, perturb motor and sensory circuits, contributing to increased pain. We are testing this hypothesis by evaluating pain behavior in newborn rabbits that experienced hypoxia in utero and by identifying changes to spinal circuitry mediating pain information to the brain. To test mechanical pain sensitivity, we placed rabbits on a mesh platform and applied von Frey filaments of increasing force to each paw until paw withdrawal was observed. We found that neonatal CP rabbits withdrew their paws in response to lower forces than control rabbits. Both motor affected, shown in dark red, and motor unaffected CP rabbits, shown in light red, exhibited increased sensitivity to painful mechanical stimulation. Next slide, please. Isolectin B4 positive nociceptors, which are pain-sensing neurons, transmit mechanical pain signals to the brain. We are using immunohistochemistry against IB4 to test whether projections from mechanosensitive nociceptors are expanded in the spinal cords of CP rabbits. Our preliminary qualitative results suggest that this is indeed occurring. In summary, we have found that neonatal CP rabbits were more sensitive to painful mechanical stimulation, even in the absence of motor dysfunction. This suggests that sensory circuits may be more susceptible than motor circuits to prenatal hypoxic injury. Expansion of IB4 positive nociceptive afferents, which carry mechanical pain signals, likely contributes to enhanced pain sensation in CP rabbits. This suggests that individuals with CP may experience greater pain due to changes in sensory neuron themselves and not just as consequences of musculoskeletal abnormalities. We hope our future work will enable novel therapeutic strategies to treat chronic pain in individuals with CP. Thank you.
Thanks, Emily. That was great. Next up, we have Daniel Powell from Bowdoin College.
Hi, everyone. Today I'm going to talk about modulatory capacity, or the total number of neuromodulators in a given nervous system, and the ways in which that nervous system responds to that modulatory capacity. And so we're going to look at how that corresponds to dietary diversity in 3 species of magoid crabs. And in particular, we're going to be looking at a nervous system that aids in the animal's digestion. So the overarching question is, will nervous systems that are given a higher number of demands going to have a greater modulatory capacity given the animal's diet? And so the 3 species we're going to look at are shown here at the bottom. The Pugetia crab only eats kelp and is closely related to the Labinia crab in the center, which is an opportunistic feeder. And so therefore, our alternative hypothesis is that modulatory capacity of a nervous system really might have to do with relatedness of species rather than the actual diet of the animal. And then our third species is shown there on the right, the Caenorhabditis crab, which is also an opportunistic feeder, similar to Labinia, but not closely related to the other two. Next slide, please. And so we're actually going to test the same set of neuromodulators in two nervous systems found in each animal. So the cardiac nervous system, which is schematized on the left, will be exposed to the same set of neuromodulators as the somatogastric nervous system, which aids in digestion. And we would anticipate that the cardiac nervous system, which controls the animal's heartbeat, would not be related to diet the way that the somatogastric nervous system might be. Next slide, please. And so what we see up top are example traces of the animal's heartbeat in response to dopamine and CCAP in blue. And on the right, response to the neuromodulator oxytremerine to the somatogastric nervous system. And then below are summary tables of responses to both nervous systems in all three species. And so what we can see on the left is the cardiac nervous system of the Puget, the kelp-eating crab. It's actually closest to the Caenorhabditis, the unrelated opportunistic feeder. And that the Labinia crab actually responds to the fewest number of neuromodulators. And so this doesn't seem to align with diet whatsoever. Whereas the somatogastric nervous system, which again helps aid in digestion of the animal's food, seems to correlate with dietary diversity, and that the Labinia crab and the Caenorhabditis crab, which are both opportunistic feeders but unrelated to one another, seem to have similar modulatory capacities. And in fact, each of these nervous systems responds to all of the modulators that we've subjected it to. And then the Puget crab, which only eats kelp, responds to the fewest number of neuromodulators. And this seems to support our hypothesis that the demands placed on a nervous system correspond with the modulatory capacity of that nervous system. Thank you for listening.
Great, thank you, Daniel. Under time, that was awesome. Okay, next up we have Karen Loeb from Brown University.
Hi there, my name is Karen Loeb and I'm a second-year medical student at Brown University who will be presenting on a study entitled Analysis of Levetiracetam Side Effects Leading to Discontinuation of Therapy and Role of Vitamin B6 in Pediatric Epilepsy. Levetiracetam is an anti-seizure medication that's used to treat both focal and generalized epilepsy. In children, it's notorious for having behavioral side effects, such as aggression and irritability, which might lead to discontinuation of the medication. In these circumstances, when patients present with side effects, their provider may choose to prescribe vitamin B6 to help ameliorate them. However, the literature is mixed on whether vitamin B6 has a protective effect against these side effects. So the goal of this study was to analyze risk factors leading to levetiracetam discontinuation and the role of vitamin B6 in altering this risk. Next slide, please. This was a retrospective chart review of 241 children with epilepsy who have taken or are currently taking levetiracetam. You can see some of the clinical measures that we extracted from the chart above. The pie chart below shows the distribution of side effects among the third of patients who experienced them. The most common side effects were behavioral in nature and included aggression, irritability, restlessness, and nervousness. Of note, almost over 60% of patients who did experience side effects discontinued the medication. The table on the right shows the key findings of the study. There was no significant difference in the mean levetiracetam dose between the groups, but there was a significant difference in the amount of B6 that patients were taking, and those patients taking B6 showed behavioral improvement. Next slide, please. The three main takeaways I'd like you to have from this presentation are that over a third of patients on levetiracetam reported side effects, and this is higher than the 25% which is reported in the literature. The second is that initiating B6 helps to reduce the behavioral side effects associated with levetiracetam, and this adds to a growing body of literature that taking B6 may protect against these side effects. So hopefully providers could use this information and prescribe levetiracetam and B6 at the same time. Lastly, there's a dose-dependent relationship with levetiracetam side effects in B6, such that patients with side effects were prescribed significantly higher doses of B6 than those without side effects. And this could help physicians have a better understanding of how to dose vitamin B6. Some future analyses we intend to conduct are reported below, and a special thanks to the collaborators that I worked with on this project. Thank you so much for your time and have a great afternoon.
Thanks, Karen. That was great. Next, we have Yuxin Zhao from the University of New Hampshire.
Hello everyone, my name is Yuxin Zhou. I am in Chen Lab at UNH. Our lab is funded by UNH COBRE program, and we are focused on neuronal primary cilia and memory. Memory is believed to be encoded by separately distributed neurons in memory-related region. However, it remains to be established how memory-eligible neurons react during learning to form and retrieval a declarative memory. Here, we utilized a deep brain calcium image system to directly visualize the real-time neuronal activity in free-moving animals and subjected to a tree-sphere conditioning. We find that the primed neuron form burst synchronization to form and retrieval a tree-sphere memory. In further study, we focused on how neuronal primary cilia affect memory formation and retrieval. We have a new cilia ablated animal models. Primary cilia are microtube-based sensor organelles present in most cells. The deflection of neuronal primary cilia will lead to many diseases like intellectual disability and cognitive dysfunctions. Next, please. We find the overall activity of hippocampal neurons show a right-skewed log-normal-like distribution. A portion of primed neuron with high activity will play important roles in memory formation and retrieval, whereas most neurons remain in silent. In our cilia ablated model, which is shown in green in this figure, we can see this overall neuronal activity is significantly reduced by cilia ablation. In the control animals, the activity pattern of these primed neurons is modified from initial randomly activity to a highly coherent burst synchronization. In order to form this memory, and in recall test, the same population of primed neuron is changed quickly from random activity to burst synchronization in order to retrieve memory. Next, please. Compared to the control animals, cilia ablation drastically suppresses the burst synchronization following field conditioning. The control animals generally display a strong and tone-responsive synchronization among their primed neurons, whereas the cilia ablated model only show weak coherence. The animal behavior test also showed the cilia ablation significantly reduced the animal field response. This line of research will advance our understanding of neuronal primary cilia and memory. Thank you for your time.
Thank you very much. That was really interesting. Next, we have Amanda Sidwell from the University of Vermont.
Good afternoon. My name is Amanda Sidwell. I look at perivascular tau and cerebrovascular function after blast traumatic brain injury, or TBI. TBI is a leading cause of mortality and morbidity that currently has no universally effective treatments. Military personnel are at elevated risk of blast TBI due to the increased use of improvised explosive devices, with over 16,000 TBI reported in the first quarter of 2020. This specific type of brain insult leads to complex downstream systemic and local effects as seen at left. We focus on primary barotrauma injury and want to understand how TBI alters cerebrovascular endothelial cells to disrupt regional control of blood flow in the brain and neurocognitive function. Neurovascular coupling is the mechanism that links transient neural activity to subsequent changes in cerebral blood flow. On the right is a representation of what happens when the protein tau is released from a ruptured microtubule into the extracellular space around a blood vessel. My research question is whether this early accumulation of perivascular tau plays a role in impaired neurovascular coupling after TBI. Next, please. Our critical first steps were to establish a survival model of blast TBI shown in the top right with impaired functional hyperemia, which is the neurovascular coupling mechanism I mentioned before. We also needed to characterize neurocognitive defects, which at the bottom right are shown to be significantly increased after 24 hours after injury. Cerebral capillary endothelial cells have potassium inward rectifier channels, known as KIR channels. Neurons and blood vessels communicate in the brain using potassium and calcium. In the graphic below, potassium is released from metabolically active neurons as a result of action potentials. KIR channels sense this elevation in extracellular potassium, and this results in a retrograde hyperpolarizing signal that propagates from the capillary endothelial cells upstream to arterioles, where the hyperpolarization leads to signals to relax the vascular smooth muscle. We hypothesize that this is disrupted after blast TBI. Next, please. For our future directions, we'll use electrophysiology, pressure myography, and in vivo imaging to assess endothelial KIR channel function. First, on the left are whole cell recordings of capillary endothelial cells. KIR channel current density is markedly reduced in endothelial cells isolated from TBI mice compared to controls. Second, on the bottom right, dilations to extracellular potassium are impaired in PL arteries after injury. These are representative traces of lumen diameters from control in TBI animals. Third, on the top right is our in vivo imaging strategy. A cranial window is centered over the somatosensory cortex and imaged using 2-photon laser scanning microscopy. This is used to visualize dynamic calcium signaling in the brain endothelium. Successful completion of these goals will offer insight into the disrupted communication following blast TBI and identification of targets for this crucial therapeutic need. Thank you.
Thank you very much, Amanda. Next up, we have Sam Short from the University of Vermont.
Hi, everyone. My name is Sam Short. I'm a med student at the University of Vermont. My mentor is Mary Cushman, and my talk is titled Plasma Opioid Precursor Proenkephalin and Neurovascular Disease Risk. So neurovascular care has seen really great improvements in recent years, but efforts to better identify high-risk individuals for future neurovascular disease has really lagged behind. Cardiovascular disease is understood to be a major risk factor for neurovascular disease, and hopefully research into cardiovascular markers can help elucidate novel pathways for better clinical risk stratification, as well as the development of therapeutics in the future. Emerging evidence shows that endogenous opioids likely play a key cardiovascular role in heart, kidney, and vascular diseases. And proenkephalin is a precursor peptide to several endogenous opioids, and it's currently the preferred assay of plasma opioid levels. However, despite these prior cardiovascular findings, the association of proenkephalin and these endogenous opioids with neurovascular diseases is still unknown. Next slide, please. So to address this gap, we measured plasma proenkephalin in a large biracial cohort. Participants were then followed prospectively, both with stroke event monitoring and serial cognitive testing. And then we used multivariable models adjusted for cardiovascular risk to better determine the association of proenkephalin with stroke and cognitive impairment each. So on the right, we see proenkephalin's association with stroke risk as shown with a spline plot. So the line is the relative risk to the median concentration, with the shading being the 95% confidence interval. As you can see, this association was nonlinear, with both high and low plasma concentrations conferring increased risk for future stroke. There was up to a twofold increased risk compared to median concentration, and this relationship was independent. Also, it wasn't impacted by adjustment for cardiovascular risk, suggesting that opioid signaling may be a novel cardiovascular pathway to stroke. Risk was most prominent among white men, as shown here, and also in Black women, but not other subgroups. Next slide, please. We also examined proenkephalin's association with cognitive impairments, and as with stroke analyses, associations were strong and nonlinear with increased risk at both high and low concentrations. Risk here was most prominent in Black women with up to 6-fold higher odds compared to the median concentration. And also in white men, though is actually relevant to all subgroups. These associations, again, were not attenuated by adjustment for cardiovascular risk factors, suggesting that opioids, again, are a novel pathway to this neurovascular disease as well. So in summary, proenkephalin is a novel plasma biomarker for neurovascular outcomes. Sex and race differences were apparent, and associations were independent and nonlinear. These results suggest that endogenous opioids could be a novel cardiovascular pathway to neurovascular disease. In the future, studies should try to elucidate the pathways by which opioids lead to neurovascular disease and then explore the utility of clinical testing as well. Thank you very much.
Thanks, Sam. And next up, we have Emily Potts from the University of Rhode Island.
Hi, everyone. Today I'm going to talk to you about the impact of stress on the nuclear pore and actin cytoskeleton. So many people know that stroke leads to neuronal death, but the neurons that do survive will have a long-lasting damage, and that may be why stroke survivors have an increased risk of developing a neurodegenerative disorder. Some of the molecular changes that we know occur after stroke are rearrangement of the actin cytoskeleton, as well as accumulation of RNA binding proteins in the cytoplasm. And we suspect that these alterations are linked to the function of the nuclear pore complex. Therefore, we hypothesize that. Next slide. Hypoxia-induced destabilization of the actin cytoskeleton compromises the integrity of the nuclear pore complex, thus impacting long-term survival of affected neurons. Next slide. So first we differentiated SHSY5Y neuroblastoma cells into a post-mitotic state and exposed them to hypoxia for 45 minutes, and then fixed the cells at 0 minutes, 30 minutes, 60 minutes, 120 minutes, and 24 hours after hypoxia. Next slide. And we looked for a few different proteins. So the first I'm going to talk about is RANGAP1, which is a marker of the nuclear pore. And you can see in the normoxia control, it has a very defined ring around the nucleus. And then after hypoxia, that becomes disrupted, and that disrupted phenotype remains up till 24 hours after hypoxia. We also looked at lamin B, which stains the nuclear lamina, which is a structure that helps maintain the structure of the nucleus. And the same thing, it has that nice defined ring, and then that becomes disrupted after hypoxia, but begins to recover at 24 hours. And both of these suggest a mild lingering defect in the nuclear lamina and the nuclear pore complex after hypoxia. We also looked at RAN, next slide, which is a protein that is essential for nucleocytoplasmic transport. And this, you can see that the RAN, there's a higher localization in the cytoplasm after hypoxia, which could be a consequence of the hypoxia-induced nuclear pore defects. Next slide. And in conclusion, hypoxia induces mild persistent changes to neuronal structure and function, suggesting a novel molecular link between hypoxia and neurodegeneration. And in the future, we hope to expand our analyses to include cell survival and other markers of cell stress in iPSC-derived human cortical neurons. We also hope to utilize this model system to test small molecules targeting the crosstalk between actin and the nuclear pore as novel therapies to prevent neurodegeneration. And I want to give a special shout out to Tinu, who was a SURF student who helped with these experiments. Thank you.
Thank you very much, Emily. Next up, we have Megan Tamash from the University of Maine.
Hello. Today I'll be discussing chronic pain-induced impairments to cognitive flexibility. So chronic pain is a prominent public health issue, and in addition to its well-characterized sensory components, it is also associated with impairments to psychological and cognitive function. One major cognitive impairment associated with chronic pain is impaired cognitive flexibility, or an impaired ability to update decision-making in response to changing contingencies. The impairments to cognitive function that are associated with chronic pain contribute to the disease's disabling impact, but their underlying mechanisms remain largely unexplored. Next slide, please. In our lab, we utilize the well-established model of chronic neuropathic pain called the spared nerve injury, or SNI model, which recapitulates the central characteristics of chronic pain. We use mice in our lab, and through exposure of the left hind sciatic nerve and ligation of 2 of its 3 branches, this is how we establish the SNI model. As you can see, SNI animals data are shown in red, and these SNI animals display persistent mechanical hypersensitivity, even 3 to 5 weeks post-surgery, when sham animals, those who have had their sciatic nerves exposed but not ligated, return to their baseline. This divergence, highlighted with the gray bar, is indicative of chronic pain, and this is the time point at which all of our experiments will take place. We assess cognitive flexibility with a reversal learning task in a T-maze assay, in which animals are trained to receive reward in one arm of the maze before it's switched to the other side. As you can see, chronic pain does not significantly influence initial acquisition of learning, but when tasked with this change in contingency, the SNI animals are significantly less successful than their non-surgerized littermates, suggesting impaired cognitive flexibility. And so together, this shows that SNI provides a model that allows us to study the mechanisms underlying chronic pain-induced cognitive flexibility impairment. Next slide, please. In the interest of time, skipping over a lot of background here, but we are interested in the nucleus accumbens, shown here in a coronal slice, as it is a major regulator of reward-motivated behavior and has been implicated in both chronic pain and cognitive flexibility. We are particularly interested in a cell type called cholinergic interneurons, which provide the sole source of acetylcholine to the nucleus accumbens. Patch clamp electrophysiology revealed a hypercholinergic phenotype in SNI animals, as you can see with the red traces on the right, where spontaneous activity levels were almost 3 times as fast as that as non-surgerized controls. And this is interesting because human imaging data show an inverse relationship between accumbal acetylcholine levels and cognitive flexibility. Therefore, our ongoing hypothesis is that chronic pain increases cholinergic signaling in the nucleus accumbens, thereby decreasing cognitive flexibility and contributing to impaired cognitive function. Ongoing work aims to identify the mechanism underlying this hypercholinergic state, as well as evaluating its causal role in impaired cognitive flexibility. Thank you.
Thank you very much, Megan. Next up, we have Nick Cottam, from Delaware State University.
Hello, everybody. Thanks for coming. Today, I'll be talking about my lab's exploratory research regarding the use of magnetic resonance imaging to study the role of the cerebellum in spinal muscular atrophy using the SMA Delta 7 mouse line. So what is SMA? SMA is an autosomal recessive disease that affects 1 in every 6 to 10,000 individuals at birth, making it the leading genetic cause of infant mortality. The disease is best defined as a motor neuron dysfunction due to depletion of ubiquitous survival motor neuron protein, SMN, which leads to the degeneration of motor neurons in the anterior horn of the spinal cord, which projects to neuromuscular junctions throughout the central nervous system. Now, there are effective treatments for SMA, but not all patients respond to them, and their long-term effects are unclear. Additionally, a growing number of studies support that SMA is actually a multi-system disease. And whereas the spinal cord has received much attention, the rest of the CNS, and particularly the cerebellum, haven't. The cerebellum is the locomotive hub of the brain, and widespread abnormalities have even been reported in the cerebellum of SMA patients. So we set out to develop a deeper understanding of the mechanisms behind SMA disease pathology across the CNS, starting with the cerebellum. And we hope to open the door to novel and more comprehensive treatment strategies. Next slide, please. So our research question is simply as follows. Is what we're seeing in the cerebellum a result of independent mechanisms, or is it a result of understood SMA pathology in the spinal cord? To address this question, we first acquired T2-weighted images of ex vivo mice brains from MRI scans and used them to acquire volumetric measurements of the whole brain and the cerebellum. We found that the cerebellar growth is significantly reduced in SMA-affected mice, and the size of the cerebellum in late-stage SMA mice is differentially affected compared to the whole brain. To further define SMA disease pathology in the cerebellum, we then studied the tractography using diffusion tensor images to quantify white matter pathway abnormalities in SMA. So we wanted to quantify the level of disorganization we were seeing in the cerebellum, as you can see in the bottom left. The one on the right is SMA, the one on the left is control, a little bit of a formatting thing. So we used the following tract classifications based on what was being reconstructed: superior peduncular tract, medial peduncular tract, inferior peduncular tract, intracerebellar connections, and below there also cut off is where we saw no tract being formed. These classifications are determined mainly on where the tracts pass through and where they terminate. Next slide, please. We found that the inferior peduncular tracts and intracerebellar connections were among the most significantly diminished tracts due to SMA in the mice brain scans relative to wild type. So from this data, we concluded that the cerebellar axonal development and concomitant white matter pathways are affected by SMA, and a better basic understanding of these neurobiological abnormalities of SMA may lead to novel and more effective avenues for treatment. In the future, more data is necessary to confirm whether these effects are just downstream pathologies or if they play an important role in disease development. So we're currently analyzing data on similar analyses in P3 and P4 mice brains. We're using slice immunohistostaining and also looking at common DTI metrics such as fractional anisotropy and various diffusivities across regions of the cerebellum. Thank you.
Thank you, Nick. Next up, we have Emma Noel from Bowdoin College.
Hi, my name is Emma Noel, and I work in the Honeycut Rat Lab at Bowdoin College. My project is titled The Impacts of Early Life Adversity, or ELA, on DNA methylation and behavior. ELA, such as childhood abuse, neglect, or other forms of trauma, has been shown to have profound behavioral and neurological effects later in life, including increased risk for mental illness. However, the nature of this biological pathway remains largely unknown. Next slide, please. Studies have suggested that epigenetic factors via alterations in DNA methylation could result in these maladaptive behavioral and neurological responses, as DNA methylation represses gene expression over development in response to changing environmental stimuli. Thus, ELA might affect DNA methylation patterns, which could then affect later life stress response. My project with the Honeycut Rat Lab looked at the effects of ELA on later life behavior and neurology in hopes of characterizing predictive biomarkers for mental illness. I used maternal separation, which is a translational model of caregiver deprivation, and then tested both male and female rats at 2 different developmental time points. I hypothesized that ELA-exposed females who translationally have earlier onset for anxiety-related disorders would show more anxiety-like behavior sooner than males. Preliminary results show that at late adolescence, both ELA males and females have more anxiety-like behavior compared to control animals. Further testing needs to be done to confirm these results. Next slide, please. Neurologically, I hypothesized that ELA would lead to an increase in DNA methylation in brain areas associated with anxiety, thus leading to a decrease in gene expression within those areas. I'm looking at the PSC, BNSG, and BLA, which are all brain regions associated with anxiety, as pictured on the right and highlighted in green. And we'll analyze the brains using both immunohistochemistry and ELISA assays. I'm using both as immuno can show overlap between parvalbumin-containing interneurons and methylation markers, whereas ELISAs can quantify global methylation within specific brain regions. Parvalbumin is a calcium-binding protein implicated in the regulation of anxiety-related pathways, and overlap with methylation may suggest an abnormal mechanism for abnormal functioning following ELA. This analysis will take place in the fall. Overall, maternal separation translationally models caregiver deprivation, which has profound effects on brain development and maturation in children. A current example of this is the US-Mexico border crisis, in which migrant children are being separated from their families and detained with limited access to necessary resources. Additionally, mental health disorders are increasingly common worldwide, with limited viable treatment options for many people. My work hopes to make strides towards individualized treatment plans by characterizing biomarkers that may eventually be targeted in translational preventative treatments. Thank you so much for listening.
Thank you, Emma. Next up, we have Sobi Sobolewski from Bates College.
Good afternoon. The title of my project is called A Data-Driven Classification of Accessory Olfactory Bulb Mitral Cells. Neuroscientific inquiry is often guided by the principle of biological form suiting function. The human brain is comprised of 86 billion neurons that guide complex emotions and behaviors via stimuli that are obtained from the environment. Olfaction is of particular interest because the process of interpreting volatile odors into tangible sensations is still not well understood. The mouse olfactory bulb, specifically the accessory olfactory bulb, is a model organ for studying olfaction due to its size and location in the olfactory bulb. The main idea behind this project is determining if there are neural circuits working together that can be defined by morphological subtypes of the main neurons within the olfactory bulb, the primary structure involved in olfaction. So what do we mean when we say morphological subtypes of the main neurons within the olfactory bulb? We're reconstructing specific neurons called mitral cells within a specific area within the olfactory bulb called the accessory olfactory bulb and seeing if and how the structure of these neurons varies. As you can see here in the figure on the right, we're reconstructing these cells in the mitral cell layer, MCL, that receive input from glomeruli. Glomeruli serve as the anatomical pathway from the nose to the olfactory cortex where odors are processed. As you can see here, the circuitry is complex, and this project attempts to untangle the meaning of some of this. Next slide, please. We can reconstruct these neurons through a whole cell patch clamp electrophysiological experiments. EPHIS is quite intense, taking a thin glass needle, also called a recording electrode, and inserting it into a cell, also called patching a cell. By patching cells, we can fill the cell with a fluorescent dye and a chemical compound that labels the cell we're interested in. After we fill the cell, we can reconstruct it in a three-dimensional space via a neuron tracing software. We can then run the reconstructed cell through a special computer program in MATLAB that turns all of the cell's structural features into numbers, which we can then analyze to see which features we want to prioritize in our analysis through a method called principal component analysis. Finally, we can take the reconstructed cells and run them through a machine learning algorithm and classify them on the basis of their structure. Next slide, please. This project requires obtaining a rather large data set to analyze correlations between morphological features, which is why we can't yet make any reasonable conclusions. It's also important to note that the majority of the summer was spent learning how to patch cells, with the first cell getting patched roughly 1 month ago. Since then, we've patched a total of 8 cells. Analyzing neuronal morphology enables us to answer questions such as how does soma and dendrite size influence connectivity with other glomeruli, or what do the other morphological features look like for mitral cells projecting to a singular glomerulus versus many glomeruli, and how do glomeruli sample? Thank you.
Thank you, Sobi. Next up, we have Joey Gallen from Rhode Island College.
Thank you. Hi, my name is Joey. I'm a recent graduate from Rhode Island College of Biology. So across the globe, nations are progressively adopting a modernized Western diet consisting of high-fat, hyper-palatable foods. This, combined with an increasingly sedentary lifestyle, has led to an increase in the prevalence of obesity, metabolic syndrome, and associated disorders. So there are 2 main central pathways of obesity that are described in the literature, one being hedonic obesity, which is driven by pleasure and reward pathways. The other being homeostatic or metabolic obesity, where the set point body weight is actually increased to a higher level. The sex-specific consequences of a fatty sucrose diet on neurobehavioral, hedonic, and homeostatic pathways are not fully understood. Therefore, the goal of this research is to discern those sex-specific effects so that more targeted therapeutic strategies can be developed to improve patient outcomes. So we hypothesized that rats fed a hypercaloric, high-fat diet would gain more weight, exhibit anhedonia, and have increased levels of anxiety compared to their control diet counterparts. And we have also hypothesized that these differences would be more pronounced and reach significance faster in the females based on the literature. Next slide, please. So male and female Sprague Dawley rats were used to model diet-induced obesity. They were maintained on either a high-fat, moderate sucrose diet, so 45% fat, 17% sucrose, or standard chow, so 10% fat, 0% sucrose, for 12 weeks. And anthropometric and neurobehavioral tests were performed throughout the study. So my apologies for skipping around. First, we're going to look at the right side. So in figure 2A and B, we see that males were fed a high-fat diet, gained weight more quickly than females fed the same high-fat diet, 7 weeks versus 10 weeks post-diet initiation. Additionally, we found that males, but not females, maintained on a high-fat diet had a significant change in body weight and consumed more calories in a 24-hour period, seen in figures 2C and D. And as I'll discuss on the next slide, we analyzed glycemia and serum insulin. After that, we did some hepatic tissue analysis, which I will not get into here for the sake of time. Next slide, please. So figure 3A shows that in general, males have a higher free-fed glucose level compared to females. And in B, we see that there's a main effect of diet and sex, showing that males fed a high-fat diet have significantly higher fasting blood glucose levels versus their control diet counterparts. In C, there was no main effect of fasting insulin level. However, there was a trend towards a main effect of diet. And lastly, we assessed anxiety using the novelty suppressed feeding test and found that males fed a high-fat diet have significantly increased levels of anxiety compared to their control diet counterparts. So in conclusion, females appear to be more resistant to the negative health effects associated with high-fat diet consumption, and future studies will do further tissue analysis in hepatic tissue, brown adipose tissue, and different regions of the brain. Thank you.
Thank you, Joey. Next up, we have Alsu Shagiva from the College of the Atlantic and the University of New England.
Good afternoon, everyone. The ability of navigating ourselves in the environment depends on structural and physiological properties of main hippocampal cells, so-called pyramidal cells. And it was observed previously in our lab that if those cells are missing a protein called IFT88, it affects spatial memory and learning abilities. IFT88 is crucial for primary cilia development. And primary cilia is a microtubule-based structure that is present in most of the cells in the vertebrate animals. Malfunction of cilia proteins in different cell types causes medical conditions called ciliopathies, and the degree of medical conditions can be as severe as defects in brain formation during development of embryo. So this summer, I was trying to see what are the structural changes pyramidal neurons have if they're missing primary cilia. And the results suggest that there is a loss of functional excitatory synapses due to decrease in the dendritic spine density. Next slide, please. Neurons receive input through dendritic spines, which are the protrusions from the dendrite, and the density of those spines is known to affect learning and memory abilities. So first, I just counted spines using bright field and confocal microscopy, looking if pyramidal neurons without cilia have different densities of those spines. In both techniques, the results showed significant decrease in dendritic spine density in knockout mice compared to wild type. The other way of assessing excitatory inputs in these two conditions would be by measuring electrophysiological properties of cells. Next slide, please. If we add some drugs to eliminate both excitatory and inhibitory inputs, we would be able to record the effect of spontaneous vesicle releases. And the frequency of those events, or so-called mini excitatory postsynaptic currents, usually correlates with the density of the dendritic spines, because more spines means that there is a bigger probability of cell receiving spontaneous vesicle, and that means that we will see more events on the trace. So preliminary results show no significant difference, which is, however, can be due to some technical issues we faced during the recording and low statistical power. To conclude, the loss of cilia in main hippocampal cells leads to decrease in dendritic spine density, which is a change on structural level, and that is resulting in a loss of functional excitatory synapses. And even though counting of dendritic spines showed strong statistical difference, more electrophysiological data is required to finish the analysis. Thank you for your time.
Thank you. Also, next up, we have Hannah Scott from Bowdoin College.
Hi, my research in the Horch lab at Bowdoin College focuses on the compensatory plasticity in the prothoracic ganglion of the Gryllus bimaculatus or Mediterranean field cricket auditory system. As you can see in panel A on the left, you can see a diagram of the prothoracic ganglion under normal conditions, and in B, you can see the same prothoracic ganglion following deafferentation or the loss of an ear, which in crickets is on their foreleg. And you can see that the neuron in black in panel B, which is AN2, following deafferentation crosses the midline, a boundary it normally respects, and forms functional connections with afferents from the contralateral ear, allowing it to undergo compensatory plasticity and continue to aid in the cricket's hearing. However, our research has found that immediately following deafferentation and prior to when AN2 has formed these new connections, it shows a novel electrophysiological response that isn't anticipated given what we know of the electrical circuit of the auditory system, a schematic of which you can see on the right. So specifically, what I'm looking at is what exactly this response is and where it's coming from, because the fact that it exists indicates that we may not know as much about the circuit as we previously thought, indicating that we may not fully understand the plasticity as we had previously thought. Next slide, please. Again, on a very broad sense, our research is looking at how does the electrophysiology of a plastic neural circuit change after injury and using the Gryllus bimaculatus auditory system to investigate this. The procedure we use is primarily exposing the prothoracic ganglion, which you can see circled in green, and then inserting a microelectrode into the main dendrite of AN2 and recording from it while stimulating it with high-frequency sound stimuli, because AN2 responds specifically to the high-frequency stimuli produced, or the high-frequency sounds rather, produced by predatory bats. Then, following recording for 30 minutes, we use cold deactivation as a simulation of deafferentation, record for 30 more minutes, and then warm the ear back up and record for another 30 minutes. We use 3 parameters to measure our results. The first is the difference in onset of time between the sound stimulus and AN2 firing. The second is the number of spikes per AN2 burst, and the third is the frequency of those spikes. We perform an across-cricket analysis as well as a within-cricket analysis to measure these results. The across-cricket analysis didn't show all that much, which is unsurprising given the fact that previous research has shown that crickets individually respond very differently to sound, making it hard to see trends across crickets. However, the within-cricket analysis showed much more specific results as to this electrophysiological response. Next slide, please. So here you can see 2 different crickets for the within-cricket response. One is on the left, the other is on the right. And these findings show that during cold deactivation, which is depicted in blue, we see an increase in delay time variability, a decrease in spike number, and a decrease in spike frequency. Additionally, we see a partial restoration of these characteristics after warming, but only in response to the more intense of the 2 stimuli. In the future, we'd like to use this characterization to really determine where this electrophysiological response is coming from. Thank you.
Great, thanks, Hannah. And I think that is going to wrap up our lightning talks. So thanks for a series of great talks. We're now going to open it up for questions. So if anyone has any questions, feel free to put them in and they will be relayed. For those of you who spoke, and if you have any questions, feel free to put them into the chat.
Hi. Also, this is Samantha Medic here. As we're waiting for some questions to come in, I want to thank everyone who participated in all the lightning talk sessions today. If you were in this track with us from the tech to the psychology to the neuro sessions, we really appreciate you being here. We appreciate all of the presenters and your hard work and your dedication, both with respect to the research you're personally doing, in addition to your presentations today, especially in the middle of what I imagine is a very busy summer for all of you. I'd like to thank our moderators from all the sessions today for their time and effort in helping us put together these talks for you. And last but not least, I wanted to let you know that if you did happen to miss some talks or you want to go back, we are going to have the recordings available on the website. In the same where you had the join button in the agenda, there'll be a link there with the recording. So if you're wanting to go back and watch some of these talks later, they will be available for you. And last but not least, if any of the judges just happen to be watching, because we have awards that we'll be giving for the lightning talks, please make sure that you go to the Google shared sheet for the judging and fill out your scores as soon as possible, because we actually are going to wrap those up pretty quickly. And if any of you presenters have any questions about that, feel free to reach out to me. I don't think we have had any questions come through for either Hubble or the chat. So I think I'm going to wrap things up. Amanda, did you have anything else you wanted to add?
Nope. I have my own question actually for Megan Tamash, if she's still here.
Yes, hello.
Hi. I have a question about the acetylcholine. And I was wondering if you've looked if there's any compensatory changes in the receptors.
So we haven't gotten there yet, at least. I'm going to be doing more of synaptic input up in the upcoming months as I wrap up this current data set that I have. And I know we are also working on some chronic fiber photometry to measure actual acetylcholine levels in vivo while the animals are performing these behavioral tasks. Does that answer your question?
Yeah, that's good. Thank you.
Samantha, excuse me, this is Samantha here again. We actually did have a question just come through on Hubble, and this is our question app, and this is for Alsu Shagiva. So just so you know, this question's coming towards you. And the question is, what is the age of the mouse you used for density quantification?
Yes, so animals were from 6 to 8 months old.
Nope. Okay, thank you. Easy enough to ask. So I didn't see any other questions coming through, so I think I'm going to wrap this up for today. Feel free to reach out if you have any questions or anything you need to follow up on. Hope you have a great rest of the day.